So in this video, I'll be talking about the structure of RNA. I already made a video on the structure of DNA, so make sure to go check that out before you check out this one. So first off, I want to start off to talk about what RNA is, because a lot of people don't know what the difference between RNA and DNA is. So I'm going to try and explain this using a kind of story, kind of a image representation. Okay, so we know that the alphabet has around, I think it's 26 letters, I'm not sure, but the point is we can have a language just by having a certain amount of letters, and when we arrange them in different orders, what we get is a different story, right? Or a different word, or a different meaning. So when say we have the book Harry Potter, and we rearrange the letters of this book into a different order, we may get something that has no meaning, or we may get another meaning, a different story, right? So, as I said in my previous video, DNA is similar to that. It has its own alphabet of 4 letters instead of 26 letters. So, DNA and RNA have their own alphabet of 4 letters. And if you rearrange these letters, you can get a different organism, a different characteristic, a different behavior, okay? So you can kind of imagine that DNA is also like a book. And if each person has their own individual book, okay? They have a different way that they are. And if you change something about their DNA, they could become different, okay? And that's when DNA mutations come in. But back to DNA and RNA. So DNA, you can kind of think of as the book. The entire book. You can think of DNA as the entire book. So all your information is DNA. Everything about you, your instruction manual, what makes you you, that is your DNA. Everything. So, but in RNA, you can think of as only a single page of that book. Okay, so if DNA is the entire book, RNA would be a single page from that. Okay, and I'll try and elaborate on this to make it make sense. So you know how the DNA is located within a cell, specifically within the nucleus, and the nucleus is surrounded by a membrane, right? It's surrounding it, so the DNA can float around in your cell. Now pretend this door is that membrane, and this door is locked, okay? So nothing can pass without specific permission, okay? Now, we know your DNA is, let's pretend this is the inside, this side of the door is the inside of your nucleus. So we know your DNA is located there, okay? Everything about you. Now, to be able to let something happen within the cell, you need to first go to the nucleus to get the information. Because this is your DNA is like your instruction manual. Without your DNA, you don't exist, right? So to make modifications inside the cell or inside your body, you first need to access the nucleus for information, okay? So your nucleus has all information. So, but the issue is, say you want to change something within your cell, and you want to find the instructions now to do it. You have to come into your nucleus. So here you come into the nucleus, and say you're trying to find the specific information. Obviously, the specific information you're trying to find is not going to be the entire book. It will be maybe one specific section of the book, right? So you go in, your body will go in and find that specific information. So say, for example, you fell and you're hurt, and your body needs to replace your skin cells because you have now a scab. So it's going to come into your nucleus, find that specific page, that instructions on how to fix it. And so once it finds it and knows how to do it, it can't just take the entire book out of the nucleus because it doesn't fit. Think about it. If this door is locked, how are you going to fit this book under the door? You can't, right? So you're going to have to do something else. The cell does something else because it can't bring out the entire DNA to go fix an issue. So what it does is it finds a specific page, like I just mentioned, but it can't just rip out the page. For example, say it finds the page, it rips it out. Now it's good because now it actually fits under the door. But the problem is, now that you ripped it out of your DNA, next time you have a problem and your cell comes to try and find the solution, it can't find it. It's not there anymore because you ripped it out. So what you have to do before you can make the sheet is go and photocopy it. So this is what this photocopier is here for. So your cell is gonna, and this process of photocopying a piece of your DNA is called DNA replication. So it's gonna take the DNA, it's gonna take it to the photocopier, and it's gonna go photocopy that specific page, and it will print it out, and that specific page that you're getting is RNA. So like I said, RNA is one page of your DNA, a replicated page or a replicated section of your DNA. Now, all your DNA is still there because you photocopied it. So next time you have a problem, your cell can still go and find the information, but now you have that specific page and it can fit smoothly under the door outside the cell, and they can go and fix this problem. They can go to the RER to go make the proteins or make the solution, okay? And next time you'll still be able to access the information. So that's the only difference between RNA and DNA. So it's really not that hard. So just remember, think of a story. RNA is like a chapter from an entire book, whereas DNA is the entire book. And you can see how that's useful because RNA can fit under the door or through the membrane, whereas DNA is too big to be able to go through the membrane or under the door, okay? So that's the only difference. I hope you understood that. But one last thing. When it goes to photocopy that specific page to make an RNA, to make the RNA that can slip through the membrane, one slight small mistake happens. So it goes to photocopy it. Let me bring this to the front. So it goes to photocopy it, but a small mistake happens. You know how DNA has 4 letters, right? The entire alphabet. So if you open this book, this DNA book, you'll just see all the letters repeating in different orders. So A, C, T, G, G, T, C, A, whatever. It repeats in different information depending on what organism it is. But when it goes to copy that specific page at the photocopier, what happens is all the letters, remember these are all the bases or the alphabet letters of the DNA. They call them bases. Let me move this up a bit. So these are the letters that you'll see repeating randomly. The issue is when everything gets copied really well. So all the G's will stay G's, all the A's will stay A's, and all the C's will stay C's, except the T's do not stay T's. They turn into another letter every time DNA is turned into, a piece of DNA is turned into RNA. So every time one page is copied into RNA, when you see this RNA section, you will notice that if you look on the page, you'll only see GAC and no T. You'll not see any T's. Every time there was a T in the DNA and it was copied, the T turned into something else. Okay? The T turned into U. U stands for uracil. Okay? So bear that in mind. So you can see one difference already. That the difference between RNA and its function is that RNA is a page, whereas DNA is a book. Another difference is that RNA has different alphabet, different bases. It has GACU instead of GAC and T. But DNA still has GAC and T. So you can see, just remember, pretend like a photocopying error went wrong. And every time that, and I'll explain kind of why this makes sense, why there's a difference in the letters, a difference in the alphabet, just shortly. But just remember this for now, that in RNA, there's a U instead of, every place there was a T in DNA, there will now be a U in RNA. And that's how you can tell when your teachers give you a list of alphabet letters. You'll know if that is from RNA or from DNA. You just have to look for T's. If you don't see any T's, it's likely, and you see U's, then it's likely that it's from RNA instead of DNA. Right, so we're almost there now. Last few differences on the next page. I hope you understood all of this. So let's go to this. Next difference. This one you probably may have seen. If you didn't, but doesn't matter. So what is this image right here? Hopefully you know, or now you know, that this is DNA. This is exactly how DNA looks like. And you can see there's 2 layers. One on the outside and then one on the inside, and they can kind of connect there in the middle. And you call this double-stranded. There's 2 strands. So DNA is double-stranded, but this right here is RNA. And RNA looks slightly different, you can see. It doesn't look like it's like connected anywhere to another strand. It looks like a singular strand, and that's what they call it, single-stranded. You can see, single-stranded. And this makes sense because when you're copying the specific page, you're not copying everything, right? You're only copying a specific section. Okay? Lastly, you know when we looked at, if you watch my video on DNA, this should be even easier. This is how a nucleotide looks like. This is what makes up your DNA or your RNA. Nucleotides are like the smallest things that make up DNA. Okay, and nucleotides consist of the base, so the alphabet letter, a sugar, and a phosphate. Okay, but there's one important thing that you have to know right now, is that another difference besides the alphabet and the function with RNA and DNA is the sugar. The sugar is different. So in RNA, this sugar is called ribose, whereas in DNA, it's called deoxyribose. Why does this make sense? So it makes sense because ribose, if you know the name of RNA, you'll know RNA is ribonucleic acid. And ribo is the beginning of the name of RNA. And so this is the sugar that makes up RNA, ribose. Whereas if you look at DNA, this sugar is something else. It's not ribose, it's deoxyribose. And that makes sense because when you look at DNA, it will be, if you say it out loud, it will be deoxyribonucleic acid for the deoxyribose sugar. So just remember, the beginning kind of specifies the sugar. So if you know the name, you'll know that there's a difference in this sugar of DNA and RNA. Now, now I'm coming back to that point of why the bases are different. Why is there a mistake in the copying? Now, an easy way to remember that there's a mistake in the copying and the letters are different between RNA and DNA is that because there are different letters of the alphabet and you can see there's a small bonding happen here between the base and the sugar. Now, because now there are different bases in RNA, so there's 3 are the same, but 1 is different. But if we use the same sugar, it won't be able to bind to that different sugar. So you need that different base, that different alphabet letter. So what happens is you need a different sugar that has like the conformation to be able to also bind to that new sugar. So deoxyribose sugar is perfect for A, C, T, and G, but ribose sugar has a perfect conformation for A, U, C, and G. Okay, so the sugars have different shapes. RNA has ribose sugar, which is perfect for its 4 bases, and DNA has deoxyribose sugar, which is perfect for its 4 bases. So you can remember that because the alphabet is now different between RNA and DNA, you need now a new sugar that can also attach this new letter, this new base. Okay, that's it for this RNA video. This is all you kind of need to know about RNA. Everything you need to know. If you know all of this about RNA, that's all you need to know. And same with DNA. If you watch my DNA video, I will now I'll make a video that compares DNA and RNA in a summary kind of way to make everything make sense again. But it won't be in detail, so make sure to watch RNA and DNA separately. After that, I'll make questions from the IB. I'll take some past paper questions and go through them with you guys just so it all makes sense.